Usually when we think about how cancer starts, we picture a single rogue cell.
Right.
Like we imagine a cell that just suddenly decides to divide uncontrollably, right? Yeah, just multiplying rapidly until it becomes this massive physical problem. It's a very chaotic, almost, I don't know, accidental image of a biological car crash.
Yeah, and the classic textbook view really leans into that idea. It's always framed as a matter of unchecked proliferation.
Exactly.
Like a genetic typo happens, the cell hits the biological gas pedal, completely forgets where the brakes are, and, well, a tumor is born.
But what if that whole picture is just fundamentally incomplete? What if early cancer isn't about blind, chaotic division at all? Because, we have a stack of source material in front of us today for this deep dive that suggests something far more calculated.
Highly calculated.
Yeah. It centers on this groundbreaking April 2026 paper published in the journal Nature. It's titled, Early Fibrotic Niches Established Tumor-Permissive Microenvironments.
It's a fascinating read.
It really is.
Yeah.
And what this research asks is, what if a mutated cell acts less like a runaway train and more like a meticulous architect, one that's deciding to build this highly fortified bunker before it even starts to grow?
It completely flips the paradigm. I mean, the research outlines this exact step-by-step microscopic chain reaction.
Right.
Where a single mutated lung cell actively hijacks the body's natural healing system. It builds what they call a tumor-permissive niche.
And the wild part to me is it constructs this protective shell long before it turns into full-blown treatment-resistant lung adenocarcinoma.
Exactly. The bunker comes first.
Okay, so let's unpack this, because our mission today is to decode this entire process for you listening.
Right.
And to really grasp the mechanics of what's happening, we need to zoom way, way in, down into the alveoli.
Right, the air sacs.
Yeah, these are the tiny, delicate, kind of balloon-like air sacs deep inside your lungs where oxygen exchange actually happens.
And right there, nestled in those delicate air sacs, are the main characters of our deep dive. They're called alveolar type 2 cells.
ET2 cells.
Yeah, let's just call them AT2 cells. Now, in a healthy lung, these AT2 cells are incredibly important. They secrete this slippery substance called surfactant.
Which keeps the air sacs from collapsing, right?
Exactly. But more importantly, for our purposes today, they act as the local resident stem cells.
Okay, so think of these AT2 cells as like the local paramedics and repair crews of your lung tissue.
That's a great way to look at it.
So when you breathe in something harsh, say wildfire smoke or heavy pollutants, and the lung tissue gets chemically burned or damaged, these AT2 cells are the ones that jump into action.
Right. They divide and transform to patch up the injury. They are the ultimate custodians of lung homeostasis. But the entire system gets thrown totally off balance by a very specific genetic mutation.
And this is the KRAS G12D mutation, right?
Yeah.
I've seen that name pop up in so much cancer literature. KRAS is practically infamous.
Oh, absolutely. It's one of the most well-known oncogenes we have. But what this Nature paper reveals is the nuance of what that mutation actually does in its earliest stages. Because when this KRAS mutation occurs in an AT2 cell, it doesn't just trigger explosive, immediate division.
Which goes against the whole car crash idea.
Right. Instead, the mutation locks that stem cell into a highly specific transitional state. The researchers call it a DATP-like state.
DATP, let me spell that out. That stands for damage-associated transient progenitor.
Correct.
So, breaking that down for you, it's a progenitor, a stem cell, that is reacting to damage. But in this case, there isn't actually any smoke or pollutant. The mutation is faking it.
Yes. The cell is experiencing a massive internal hallucination.
Wow.
The KRAS mutation causes sustained activation of a transcription factor called NFB.
NFB?
Yeah. Now, NFB is essentially a master switch for inflammation. When it's flipped on, the cell's nucleus reads that signal and says, uh-oh, we must be under severe attack.
Even though everything outside is fine.
Exactly. So this mutant cell behaves exactly as if it is constantly repairing a catastrophic wound, even though the surrounding tissue is perfectly healthy.
It's pulling the fire alarm, but there's no fire. And because of that NFQ activation, it's stuck in this endless emergency repair loop.
And because it thinks it's managing an ongoing crisis, it undergoes a functional shift. It transforms into a central signaling hub.
A signaling hub, so it's not just sitting there.
No. A normal AT2 cell is quiet, it does its job and stays out of the way. But this mutant AT2 cell starts shouting biological orders into the microenvironment.
Like a rogue architect.
Yes. It's preparing to direct all the neighboring cells to help heal this phantom injury.
Man, that is incredibly deceptive. It's literally using the foundational logic of our own biology against us.
It really is.
So looking at this first crucial step, lung adenocarcinoma doesn't begin with a tumor mass. It begins with a lung stem cell trapped in repair mode because of a genetic lie.
Beautifully put.
Which brings up a really profound question for you listening. Have you ever thought of cancer as a wound that just refuses to heal?
Viewing the disease through that lens, it just changes everything about how we might approach it. It's not a foreign invader arriving with its own weapons. It's a corruption of our own internal emergency response system.
Okay, let's do a quick mental checkpoint here to lock in this first piece of the puzzle. I'll give it to you in bullets.
Good idea.
Bullet 1. We know that this specific type of lung cancer often originates in AT2 cells, the lung's natural repairmen.
Right.
And bullet 2.
Yeah.
We know that the KRAS G12D mutation traps these cells in a regenerative DTP-like state. The cell thinks there's an injury, flips on the inflammatory NFP switch, and turns into a rogue architect barking orders.
So the biological foundation is set, and this is crucial. An architect alone can't build a bunker.
Right. Here's where it gets really interesting. The architect doesn't pour the concrete. So how does this single solitary mutant cell actually get the rest of the healthy lung tissue to do the heavy lifting?
Well, we have to look at the communication wires, the actual molecules passing between these cells. The researchers discovered that our DTP-like mutant AT2 cell starts pumping out large quantities of a specific signaling protein. It's a ligand called amphiregulin.
Amphiregulin. I mean, that sounds like some kind of prehistoric underwater dinosaur.
Kind of does, yeah.
Let's just call it AREG.
AREG works.
So AREG is the corrupted blueprint the architect is handing out.
Exactly. AREG is the blueprint, and it specifically targets a group of neighboring cells called alveolar fibroblasts.
Fibroblasts?
Yeah. In a healthy lung, fibroblasts are the vital structural support cells. They maintain the delicate, flexible scaffolding of collagen and elastin.
That allows your lungs to stretch and compress with every single breath, right?
Exactly. You can identify these normal, healthy structural fibroblasts because their surfaces feature a specific marker protein. It's called PDGFR-alpha.
Okay, so the architect hands the AREG blueprint to the local framers, these PDGFR-alpha fibroblasts. How do they actually read the blueprint?
So the AREG ligand physically binds to a receptor on the surface of the fibroblast called EGFR.
The epidermal growth factor receptor.
Right. Now, EGFR is a kinase. When AREG binds to it on the outside of the cell, it triggers this massive chain reaction of phosphorylation on the inside.
It's changing the internal wiring.
It alters the fibroblast's internal signaling cascades entirely, and the result is catastrophic for the normal function of the lung. It violently reprograms the cell's gene expression.
So they completely change their identity. They stop being flexible scaffold builders.
Yes, they undergo what the paper terms fibrotic reprogramming. The normal PDGFR alpha marker drops, and they start expressing entirely new markers.
Like what?
You see a spike in PDGFR beta, a transcription factor called RUNX1, which shifts their lineage, and critically, a protein called ACTA2.
Okay, let's pause on ACTA2 because I know from reading the sources that this is a major key to understanding the physical change. ACTA2 stands for alpha-smooth muscle actin.
Yes.
Wait, if that's a muscle protein, why is a lung support cell suddenly manufacturing it?
That is the brilliant, insidious nature of scar tissue. When a tissue is deeply wounded, you don't just need new scaffolding. You need to pull the edges of the wound together to seal it.
Oh, wow.
Yeah. Fibroblasts expressing ACTA2 become myofibroblasts. They gain the literal physical ability to contract.
They can squeeze.
They grab onto the surrounding matrix and pull it tight, churning out thick, dense collagen.
To use a real-world analogy for you listening, it's like dealing with a corrupted city zoning board.
Okay.
You submit a permit to build a nice, airy, flexible wooden gazebo.
Mm-hmm.
But the rogue architect overrides it, hands the contractor the AREG blueprint, and suddenly the builders are pouring 3 feet of impenetrable steel-reinforced concrete.
That is exactly what's happening.
You're building a dense wall of scar tissue fibrosis right around the mutant cell.
And the spatial sequencing data in the source material maps this out beautifully. I mean, within just 1 week of the KRAS mutation activation, 1 week, you can look under a microscope and see these newly reprogrammed fibrotic fibroblasts making direct physical contact with the mutant cells. By 2 weeks, almost all of the expanding circulating mutant clones are entirely encased in this fibrotic shield.
The bunker is being built concurrently with the very first cell divisions.
Right.
It's terrifying, but also, you have to admit, a dark marvel of biological engineering.
Yeah, it really is.
So let's do our second mental checkpoint here. Bullet 1, we've got the mutant AT2 cell releasing the corrupted chemical signal, AREG.
Yeah.
Bullet 2, that AREG physically binds to the EGFR receptors on our normal structural support cells, the fibroblasts. This triggers an internal gene shift, upregulating things like the motor protein ACT2, which forces them to transform into dense, contracting, scar-making cells.
Marked by PDGFR beta.
Right. And they form a physical barrier that isolates the early tumor from the rest of the lung. Think about the implications of that for a second. If you could somehow peer into your own lung tissue right now and look at a tiny patch of fibrosis, would you be able to tell the difference between a normal healing scar from an old infection and the fresh foundation of a microscopic tumor?
You wouldn't, because at a molecular level, they're exploiting the exact same biological pathways. The nascent cancer is fundamentally wearing a disguise stitched together from normal wound healing material.
The physical wall is up, the concrete is poured. But a bunker isn't just about thick walls, is it? It needs active defense.
Right. And what's fascinating here is the fibroblasts don't just build the physical barricade. Once they are reprogrammed by that EGFR binding, they also begin actively recruiting a biological security force to patrol the perimeter.
Hold on. A fibroblast is a structural cell. How is a contractor pouring concrete suddenly hiring immune cells to act as muscle?
Through a second, highly specific molecular distress signal.
Another signal.
Yes. These reprogrammed fibrotic fibroblasts begin secreting a large extracellular matrix glycoprotein called tenascin C.
TNC for short.
It's exactly fancy.
Okay, so we had AREG as the blueprint, and now TNC is, what, the distress flare being shot up from the construction site?
That's a perfect analogy. TNC is fascinating because it acts as what biologists call a DAMP. D-A-M-P. It stands for damage-associated molecular pattern.
Okay.
It floats out into the immediate microenvironment and encounters alveolar macrophages. Now, macrophages have a receptor on their surface called TLR4, or toll-like receptor 4.
Now, I know a bit about TLR4. Isn't that receptor usually used by the immune system to detect bacterial infections?
Usually, yes.
Like it binds to the outer coating of bacteria so the macrophage knows to attack?
Yes. TLR4 is famous for recognizing bacterial lipopolysaccharides. It's an alarm meant for foreign invaders. But TNC, a protein made by our own hijacked fibroblasts, has the unique ability to bind to that exact same TLR4 receptor.
Oh man, so it hacks the bacterial alarm system.
Completely.
Now, macrophages are typically the heavy infantry of the immune system, right? Yeah. They are the cleanup crew that engulfs and destroys damaged cells and pathogens.
But when TNC hits their TLR4 receptors, the macrophages are corrupted. The signal essentially tells them, hey, this isn't an invader, this is a massive tissue injury that needs protection.
Wow.
So instead of recognizing the mutant AT2 cell inside the bunker as a threat and consuming it, the macrophages undergo their own reprogramming.
Of course they do.
They upregulate a scavenger receptor called MESR1, and they physically swarm and expand around the outer borders of the tumor.
The security guards have officially been bought off.
Yep.
They are standing right outside the bunker, but they are taking orders from the architect.
And they are highly active guards. These corrupted macrophages start pumping out aggressive inflammatory cytokines.
The chemical weapon.
Exactly. The key one identified in the research is interleukin-1 beta, or IL-1 beta.
Okay, let's trace this chain for the listener, because this is getting deep. The architect signals the builders with AREG. The builders signal the security guards with TN. The guards start shouting inflammatory signals like IL-1 beta into the lung. What does that inflammation actually do to the surrounding tissue?
It creates a self-amplifying feedback loop that expands the bunker's footprint.
It gets bigger.
Yeah. That IL-1 beta from the macrophages washes over the tissue and triggers a second wave of fibroblast reprogramming. But this happens further out, at the very outer edge of the tumor boundary.
Okay.
It creates a new layer of what the researchers call inflammatory fibroblasts.
So these are different from the ones pouring the dense ACT2 concrete.
They are biologically distinct, yes. These outer layer fibroblasts keep their original PDGFR alpha marker, but they start expressing a protein called leukocorin-2, or LCN2.
What do?
Well, they don't make the dense scar tissue. Instead, their job is to sustain the inflammatory environment. essentially creating a hostile moat around the concrete wall that keeps the rest of the normal immune system confused and at bay.
Wow. Okay, so we have a multi-layered fortress here.
We do.
An inner core of dense fibrotic fibroblasts, surrounded by a perimeter of corrupted macrophages, encircled by an outer moat of inflammatory LCN2 fibroblasts, all shouting at the immune system to stay away.
It's an incredible defense system.
To help you listening visualize this incredible causal chain, I want to give you a mnemonic. Let's call it the AFM loop. A is for architect. The mutant AT2 cells secretes a reg. F is for framers. The fibroblasts receive a reg and secrete the distress flare, TNC. M is for muscle. The macrophages receive TNC, lock onto the perimeter, and release the inflammation that sustains the whole structure.
The AFM loop, I like that. It highlights the most important realization of this entire paper. Tumor initiation is reciprocal.
What do you mean by that?
I mean, the cancer cell isn't just surviving an immune response, you know? It is actively choreographing an elaborate dance between fibroblasts and macrophages to build its own protective niche.
Let's do our third quick checkpoint, bullet 1. We have the corrupted fibrotic fibroblasts releasing tensin C.
Right.
Bullet 2. TNC hits the TLR4 receptors on macrophages, tuning them from defenders into co-conspirators.
Yep.
And bullet 3. Those rogue macrophages release IL-1 beta inflammation, creating a second outer layer of inflammatory fibroblasts, locking the entire AFM loop in place.
And you know, it really reframes why late-stage lung cancers are so notoriously difficult to treat, particularly with immunotherapies.
Right, because by the time a patient is symptomatic and a tumor is visible on a scan, this multi-layered bunker is fully operational.
Exactly. The immune system hasn't just failed, it's been actively structurally locked out.
Okay, this mechanism is brilliantly complex. But I have to push back here for a second.
Sure.
Because we see this all the time in scientific literature. The sources detail how all of this was meticulously mapped out in a genetically engineered red-2-grass mouse model.
Yes, the mouse models.
And, I mean, we cure cancer in mice every other week. But mice are not humans. A mouse lung doesn't face decades of human environmental exposures. Does this elegant architectural metaphor actually hold up in human tissue?
It is the most critical question to ask when reading any biological study, and it's exactly where this Nature paper transitions from being just a fascinating biological curiosity into a genuine medical breakthrough. Because the researchers did not stop with the mouse models, they went to incredible lengths to validate this in human biology.
How so?
First, they utilized human LUAD organoids.
Organoids, these are essentially miniature three-dimensional human lungs grown in a lab using actual human stem cells. Right.
Correct. They are vastly more complex and accurate than just growing a flat layer of cells in a petri dish. And beyond the organoids, they also ran single-cell RNA sequencing on actual surgical biopsies from early-stage human lung adenocarcinoma patients.
Okay, and when they looked at the human data, did the blueprint match?
It was a near-perfect match.
Really?
Yeah. In the human tissue, they identified the exact same ARU high epithelial states. They found the same dense fibrotic networks forming, marked by those human equivalents of ACTA2 and RUNX1, physically abutting the very earliest tumor cells.
So the human architect uses the exact same biological contractors.
Yes. But the validation goes even deeper because, you know, lung cancer isn't a monolith. It isn't only caused by KRAS mutations.
Right, there are other drivers.
A massive driver of the disease is a mutation in the EGFR gene itself, specifically a mutation called EGFR-L858R. The researchers looked at that entirely different genetic subtype, and they found the exact same REG EGFR circuit driving the construction of the niche.
That is massive, because if this AFM loop isn't just a quirk of one specific mutation, but is conserved across different genetic drivers of lung cancer, it means this is a fundamental rule of how this disease operates.
Exactly. It's a rule.
And if we know the rule, we might be able to break it. If we know exactly how the communication wire works, can we cut it?
We can. And the researchers call this the ultimate therapeutic vulnerability, the kill switch.
The kill switch.
Because this entire massive bunker construction happens extremely early, before the malignant cells start their massive uncontrollable outgrowth. There is a distinct window of time where the tumor is utterly dependent on this communication loop.
It's a window where the concrete is still wet.
Exactly. If scientists disrupt that very first step, the AREG EGFR axis, the whole structure completely collapses.
How do they disrupt it?
In their human organoid models, they used an already FDA-approved EGFR inhibitor drug called gefitinib.
Gefitinib.
Yeah, and when they applied it, The cascade halted. The AREG signal is blocked.
So because the fibroblasts never received the blueprint?
They never reprogrammed into dense ACTA2 expressing concrete pores.
Because the fibroblasts didn't reprogram, they never secreted TDM.
Right. The macrophages never got called in. And without that protective niche, the initiation of the tumor just completely collapsed.
Man, think about the implications of that. This gives us a totally new philosophy for cancer prevention.
It does.
I mean, we spend billions trying to invent magic bullets that can perfectly identify and kill every single late-stage cancer cell. We wait for a massive fortified tumor to form, and then we bombard it with incredibly toxic heavy artillery.
Chemo, radiation.
Right. But what if we just snip the communication wire between the architect and the builders before the walls even go up?
It shifts the entire therapeutic focus from trying to destroy a fortified tumor to simply dismantling its environment. If you deny the mutant cell its protective niche, it remains an isolated, vulnerable anomaly, one that the body's normal immune surveillance can likely handle without heavy interventions.
So we know this exact loop happens in humans, and we know it has a built-in kill switch vulnerability if we can intercept it early enough. Which leads to a crucial question for you to ponder right now. Could the future of treating this disease look less like dropping bombs and more like cutting the phone lines at a construction site?
It's a great image.
Could intercepting this niche formation prevent lung cancer from ever progressing to those deadly treatment-resistant stages?
The data strongly suggests that intercepting the communication is far more effective than cleaning up the explosion. It's a literal paradigm shift in preventative oncology.
We've covered a tremendous amount of ground today on this deep dive. From stem cells faking injuries, to the AFM loop of corrupted fibroblasts and macrophages, to the incredible potential of an early therapeutic kill switch.
We have.
But as we wrap up, there is one final mind-bending detail from the source material that we have to talk about.
Oh, yeah. It is arguably the most hopeful finding in the entire publication.
Tell him.
So, when you examine late-stage advanced lung cancer, the scar tissue, those cancer-associated fibroblasts we've been talking about, are fundamentally hardened.
Like actual concrete.
Right. They have undergone deep epigenetic changes. They are locked into that fibrotic state permanently. But the researchers made a stunning discovery about the early fibrotic states we've been discussing today.
The ones in the wet concrete phase. They aren't permanent yet.
Exactly. They are completely plastic. They are fully reversible. The researchers found that if you block the oncogenic signaling during this early window, those dense fibrotic fibroblasts actually revert back to their healthy, flexible PDGFR alpha state.
Wow.
The macrophages stand down and return to normal patrol. The tissue actually remembers how to be healthy, the bunker dissolves, and normal lung architecture is restored.
That is just incredible. I mean, it brings us right back to where we started our conversation today. We are so conditioned to think of cancer as this unstoppable, forward-marching force, a bleak, inevitable progression of bad cells dividing until they overwhelm the system.
That's the old view.
But if early cancer relies entirely on hijacking our body's natural, beautifully complex healing response, it completely changes how we view our own biology.
It really does.
So think about this as you go about the rest of your day. If a nascent tumor desperately needs our own healthy cells to build its bunker, and if that early bunker is completely reversible, might the ultimate cure lie not in inventing increasingly toxic chemicals to kill cells, but simply in reminding our tissues how to heal correctly?
It transforms our bodies from being mere victims of cancer into the active environments that hold the key to stopping it. If we can control the environment, we can halt the disease before it ever truly begins.
Thank you for joining us on this deep dive. Keep questioning the world around you, and we'll catch you next time.